Hi everyone, today we are going to speak about the function of the antibody, and to understand that we are going to imagine that there is a wound and a virus or bacteria is trying to enter your body. So how does your immune system recognize it and stop it? The answer lies in antibodies, not just their structure but what they actually do. In the last two videos we discussed the basic and advanced structure of antibody. Now let's just discuss what is the actual function of the antibodies. So first is the neutralization. The first and most straightforward function of antibodies is neutralization. Antibodies bind directly to viruses, bacteria and toxins and block them from attaching to the host cells, as you can see on the screen in this diagram. So for example, many viruses must bind to specific receptors on our cells to actually get entry into the cell, because the cell membrane is not permeable to foreign material, so they need to bind to our receptors. When an antibody binds to that viral surface protein, entry is blocked. Here the FAB region binds the antigen, but nothing else is needed. The pathogen is neutralized simply by being blocked from getting itself attached to that specific receptor. Next is called opsonization. The second major function is opsonization. In this process, antibody coat the surface of a pathogen. This coating acts like a tag. For example, a tag which tells our immune system that this is a pathogen and the immune system should eat it, or in other words, should engulf it. So here, the pathocytic cells like macrophages and neutrophils, they actually have the FC receptor. They can bind to the antibody and engulf it. They recognize the FC region of the antibody and engulf the pathogen. So antibody do not kill directly here. They make pathogen more visible to the immune cells. Next is the complement activation. Antibodies also activate complement systems, as you can see on the screen, specifically the classical pathway. When IgG and IgM binds to an antigen, the FC region can trigger a cascade of complement proteins. This can then lead to inflammation, opsonization, and sometimes direct lysis of the pathogen. This is a beautiful example of how adaptive immune activates innate immune system. Next is agglutination and precipitation. Because antibodies have at least two antigen binding sites, as you can see on the screen, they can bind multiple antigens at once. This causes agglutination, clumping of cells like bacteria, and it makes a very solid big clump of these bacteria. For soluble antigen, this results in precipitation. These processes make pathogens easier to remove by phagocytic cells and immune clearance mechanism, same like engulfing it and destroying the bacteria. Next is something called antibody-dependent cellular cytotoxicity. It's also known as ADCC, which is much easier to pronounce. Antibodies bind to infected and abnormal cells. As you can see, natural killer cells recognize the FC region via FC receptors and release cytotoxic molecules. The target cell undergoes apoptosis, as you can see on the screen. Here, antibodies act as a bridge between infected cell and killer cells. So, in conclusion, antibodies protect the body by neutralizing pathogen, tagging them for destruction, activating the complement system, clumping antigens, and directing immune cells to kill the infected cells. And here's an image to summarize all the functions of the antibody at once. So in the next video, we'll explore antibody classes IgG, IgA, IgM, IgE, and IgD, and we will speak about why each one exists. This video will be part of a bigger playlist for a crash course in immunology, and please subscribe and like the video and save this playlist for more videos like that. Thank you. Bye.